So what I want to tell you about today is obviously related to cryo-ET. How do we actually look at proteins in the native environment inside the cell? So during the, we have a fifth million process in between, and then we can take that to the microscope. We can tilt the specimen and take it to the series and produce a three-dimensional view of that particular area of the microscope, of the specimen. So I'm going to give you some good news and some not so good news. So I'm going to cover both sides of the spectrum. So we've been able to make advances in, improving the throughput of this technology, but there's still, very big challenges ahead that we need to, to overcome. And I think, presenting some of those challenges to this type of audience, could really, I think the field could really use, the contributions from some of the applied math people in the audience. So what do we get? When you look at those 3D images, this is an image from Julia Mohammed. Essentially, you get sort of like intermediate resolution, like 20 to 30 Armstrong, 3D picture of this particular area of the cell, where you can observe, in this case, there's some of the nuclear pore complexes in here. Then you have filaments, you have ribosomes. But the bottom line is that you get this information about where all the components are situated within the image. And then you also have proteins are actually present in multiple times, and that's being cryo-EMers. We can actually go ahead and take advantage of that repetition and average them in 3D, and that's what we call sub-tomogram averaging. But the reason I'm saying this is that there's really two objectives for why you want to do cryo-electron tomography, and one is to get just the overall structure to see where the components are situated with respect to each other, and then the other one is get high resolution representations of the things that actually repeat themselves using averaging. So one thing that came up a few times during this meeting is that these data sets, a single tomogram can actually be quite big, about 60 gigabytes roughly, just to to give people an idea. And actually, in terms of data collection, they take a long time to collect compared to single particle, right? So it's about between 20 and 30 minutes, and it depends on the microscope, the camera, and the conditions. But just to get an idea, it takes that long to get a single tomogram, right? So if you're trying to do subvolume averaging, right, in single particle, we're used to looking at, 8,000 micrograms, things of that sort, and then the samples are actually purified, right? So here we're looking in situ, there's no purification, so there's everything the way in the physiological concentration, and taking a single one of those takes half an hour. So actually, if you go and look into the Empire database, how big are these data sets? So a data set is typically multiple tomograms, right? And if you look at the size of those data sets, most of them are actually less than 15 tomograms, right? So a single data set is only 15 or less tomograms. And then we have some that go to, there's 25, some over 100, there's a few. And then there's one outlier, which is 250. But the point is that these data sets are actually small, almost too small to be able to do any kind of meaningful averaging, right? And the reason for that is that data collection is very, very slow. So we need to make those data sets larger. That's what I'm trying to advocate for here, because we know from single particle that the more particles you have, the better, and especially when you're looking in situ, where you have maybe more variability, you want to be able to have even more particles than you do in single particle if you're going to be successful. So the first reason we need to get more data, obviously, is so we can increase resolution. So this plot here is showing the difference in resolution between tomography and single particle. There's obviously a gap, and this came up many times during the meeting. Maya was mentioned this morning that it's really a challenge if you have low resolution. In this case, about 20 Armstrong, it's really difficult to fit any kind of meaningful atomic models. So obviously, we want to be somewhere closer to single particle cryo-EM, where we actually have more features and make the modeling a lot easier. So that's, I think, one of the things that will contribute to closing that gap is to actually be able to collect data faster and have more data so you can do, you can average over more particles. Obviously, the second reason, as I mentioned earlier, is that these samples, just by nature, they're going to be more heterogeneous, right? So we're going to need to have accumulate more particles so we can sort out, different conformations. And this image is also taken from Zhu et al. From this year. And this is for the ribosome, but for other proteins, you can also expect that there's going to be a lot of variability when you're looking at those in situ. So you want to be able to have enough particles so you characterize them in 3 dimensions. So when you go on to the microscope and collect till series, there's a couple of options these days. So one is like the high quality one, but it's very slow. So you collect each till series takes 20 to 30 minutes, but you're guaranteed that your data is going to have good quality. If you wanted to sort of like cut some corners, you can actually use this very clever technique where actually as you're tilting the stage, you're actually collecting a movie, right? Because we can collect movie frames. So as you're tilting the frames, and this is continuous tomography or fast incremental single exposure that Grant's group came up with. And you can collect data really, really quickly. You can cut down by a factor of 10, and then in 2 to 3 minutes or even less, depending on the conditions, you can actually get a till series. Right? The problem is that because the stage is moving, you're going to have a lot of drift as you're acquiring the movie because the stage is, it's actually moving during the process, and that's going to actually hurt you in terms of high resolution, right? So the data is not going to be as high quality as the first approach. And moreover, it's going to be very difficult to keep the stage on the tracking to be accurate, so you can actually track the exact same position as the stage is actually moving. So those are 2 challenges, but depending on what your goal is, if you just want to get like a rough, low-resolution overview of your sample, this may work perfectly well, and I'm going to show some examples where you can actually get even better than you can get sub-10 Armstrong reconstructions using this fast acquisition method. So we actually wanted to see if we could get the 2 things at the same time, right? So we don't want to give up the high quality data, but we want to be able to get it faster. So then what we did is we actually used a technique which is very common in single particle cryo, which is using beam image shift. So normally when you are trying to image a hole in your grid, you actually the beam comes straight down onto the hole, and you can get your image. So the sample plane is here, and you can see the beam going through there. Now, if you wanted to image a neighboring hole, typically what we do is we move the stage. So now we can fire the beam on the same spot. Instead of doing that, what we can actually use is we can actually deflect the beam using beam image shift, and then just by changing some of the magnetic fields, we can actually deflect the beam to a slightly different position, and that way, without changing the stage, we can actually take multiple projections, and we can actually select all the neighboring holes in here. And then without moving the stage, we can actually take multiple images. So essentially what we did is working in collaboration with Jonathan and Mario from NIHS. We extended this idea to tomography. So now we start with a 0 tilt, and then instead of taking a single projection, we actually take, in this case, like a 5x5 beam image shift pattern. So we take 25 positions. Then we tilt the stage to 3 degrees, and we take another 25 positions, and then we keep doing that. So in one entire tilting cycle, we actually get 25 tilt series all at once. And that comes down to about 2 to 3 minutes, again, depending on the conditions. But this is actually also a factor of 10 improvement in the speed of data collection, and we don't give up data quality, right? So we can do this at very high magnification, and we can preserve all the high resolution information, and we can do this quickly. So this work was published last year, and then we actually validated this on a test sample. In this case, we're using like this small. This is a single particle sample that we're just using for testing here, but this is a fairly small protein by cryo-Electron tomography standards. So it's 300 kilodaltons. So we were able to image at 1.37 Armstrong per pixel, which is fairly high Mach for tomography. We can do like 5x5 pattern, and in a few hours we collected 275 tilt series that we can then do sub-volume averaging and we have about 34,000 particles, and then we can get all the way to near atomic resolution, where we can see obviously side chains, and this obviously makes atomic model fitting much easier, of course. So this just proves that we're not missing on the high resolution information, but everything is preserved, and we can actually get to high resolution. Moreover, we actually did comparisons, and other people have done this as well. So if you do this properly, and you collect either tomography or single particle data, you can actually get to very similar quality of reconstructions, meaning that that gap that I talked about in resolution, we may actually be able to close that at least for some of the favorable cases. But this just shows that the technique itself is capable to getting to resolution similar to single particle cryo-EM. Now, BISECT, beam image shift electron cryo-tomography, is most useful to this type of samples. So here I'm showing liposomes. So if you have a membrane protein that you want to study in the native state, you can put them on liposomes, and you can use BISECT to really get a lot of data very quickly. We were actually able to image a sample like that, and over a weekend we got over 1,000 tomograms, which is these are numbers that obviously there aren't any existing data sets of that size in the empire and also elsewhere. So this just really allows you to collect data very, very quickly. You can also apply this to samples where you have viruses. This is a SARS-CoV-2. If you wanted to study the spike, you could actually use this to collect also a lot of data, so that you get into the numbers that you have in maybe not quite what you have in single particle cryo-EM, but a lot better than what we could do before. You can actually use this beam image shift tomography to look at in situ lamella, and there's been a couple of groups that have actually shown that this actually works. So Rado's group, there's the preprints are here, and then also Jürgen's group in Germany, they're able to do this multi-shot beam image shift tomography to actually speed the throughput. Okay, so that's all nice and fine, but the first time we ran this for 2 days and we got this many till series, we realized how big of a problem we got ourselves into. Because we just didn't know how to handle 1,000 till series, right? So the first thing that we realized is that you actually need to be able to process this till series in an automated manner, because otherwise nobody can do this manually on 1,000 till series. So obviously one of the things that we use routinely in single particle is to be able to monitor the data quality as the data is coming out of the microscope, and that's actually very useful because it allows you to assess the data quality on the fly. And then you can, decide, okay, I have enough good data, I'm going to stop, I'm going to go to the next sample. Now, we wanted to actually do something similar for tomography, but it turns out that that's actually, more difficult. There's a lot more problems that you need to solve, and if this needs to run on the fly, everything needs to be automated, right? You cannot afford to be, making changes as the data is being collected. So one of them, some of the challenges are that you actually now you need to align your till series, right? Not just your movies, but also your till series. And that has a lot of problems because you may have fiducials or you may not have fiducials, so you need to figure out how to align your data in either case. Sometimes you have this, some of the grid bars may blank the beam, and then you have some data that you need to discard, so you need to be able to do that on the fly. CTF estimation is obviously a lot more challenging because now you need to estimate tilted CTF on each of the images of your tilt series. And the way we are doing that actually depends on the tilt series alignment. So if the tilt series alignment fails, you can't really estimate the CTF properly. And then you need to reconstruct your tomograms. And one of the main challenges is that all of this actually takes computational resources. So be able, if you get 25 tilt series, all at once, you actually need to have enough resources to be able to handle all of those in parallel. So these are all practical things that you need to worry about when you're actually collecting data in high throughput mode. And if you actually don't have that in place, it can actually slow you down significantly, like it did for us. Obviously, we were not, we didn't plan ahead for that, so we just had to solve that problem on the fly. Okay, so just to give you a little bit, just one example, when you have a tilted image, obviously the defocus is going to change. with respect to the orientation, the position of the tilt axis. So if you just run like a regular CTF estimation, CTF find 4, the best you can hope for is to get 1 or 2 of the first TOM rings, but then everything else is going to be completely off. So if you were able to align your tilted images correctly, you can actually account for that change in the focus, and then you can actually see for the same image you can actually estimate the signal for much higher resolution, and you can see oscillations to high resolution, which is what we actually need to convert this into high resolution 3D structures. So we actually have, we developed a tool in the lab. So Jeff Martin is the main person behind this that actually allows us to do this on-the-fly data analysis for tomography. Right now we're doing all the first steps up to 3D tomogram reconstruction, and that has been useful enough for us. Now the main thing is you want to make sure that these tilts are going to be alignable, and you're going to be able to convert them into tomograms, right? And because that doesn't always happen. So at least you want to make sure that you can get to that. So we're getting this feedback as the data is being collected, and that has been very useful. Okay, so that pretty much covers like the first 2 steps. This is the pre-processing, so tilt series alignment and tomogram reconstruction. And now comes the fun part, right? So now comes the data processing downstream, and I broke it down into some simple steps. So the first one is particle picking. Obviously, particle picking a lot more interesting than in 2D. There's a lot more challenges, but also a lot more room for coming up with good ideas. So here I'm showing an example of an HIV virus. This is work that we did several years ago, where we actually were interested in studying the envelope glycoprotein here. So you need to. I'm going to tell you a little bit how we actually did that, and then eventually, once you know where your particles are, you can actually combine them all together using sub-tomogram averaging. At the end of the day, you get your final, hopefully high resolution structure. So I'm going to tell you a little bit about our efforts in trying to, make particle picking a little bit more automated or more, consistent with high throughput data collection. And there's many efforts in the, in this community, trying to tackle that because it's, there's a lot of solutions out there, but it's also still a lot of room for improvement. So typically, and you've seen examples of this in this meeting. One of the methods that people use is actually template matching. So template matching, you actually need to have a template, right? So that is sort of like a requirement, obviously. And it can also lead to several false positives, and it can actually take quite a long time to run because you need to do this in 3D. But for some particular cases, when you have a lot of particles in one tomogram, you can actually do this, and it works quite well. But it doesn't really scale up for when you have 1,000 tomograms, for example. So this is one example on HIV GAG. I think this was done template matching using EM Clarity. And more recently, there's obviously methods based on neural networks. Most of them, at least in 3D, they tend to be fully supervised, meaning that you need to spend a lot of time selecting your labels. And that is actually, obviously time-consuming. And if possible, you want to avoid, having to click on over, 1,000 particles on several tomograms, because that will just slow you down. And then because you have a lot of data, then these networks can be tricky to train, and it can also be time-consuming. So what we are doing, and this is work that Wendy in the lab, she's a student from computer science. So she's actually had some work on 2D, and then this is like the 3D part, which was recently published in the ECCV, I think last month. Where she's actually using this weekly supervised particle detection approach. And the whole point here is, what is the minimum amount of labels that you can get away with and still be able to detect these proteins in 3D. So I won't go into the details, but essentially, she's using a positive label approach, which is similar to what Topaz uses for 2D, that actually allows you to only click on where the particles are, and then using different strategies for consistency training and other modules, you can actually make up for that lack of labels and still be able to have very good performance. So we actually tried this on a couple of different data sets. So this is one from actually the same one I showed before. So these are purified ribosomes. This is from MPI 10304. And there is a comparison here with, so Cryo-3D is sort of like an extension of a model that uses the YOLO model that was developed for 2D. And then there was an extension done for 3D. So this is an example of a fully supervised approach where you actually need, in 3D, you need to actually label your particles in multiple slices, right? Not just one position in 3D, but you need to go through all your slices and label all your particles. So labeling is actually, annotation is actually very time consuming. And then in our approach, what we did is we started using like a fully annotated data set. And then we try to reduce the number of labels. In this case, I'm showing when we use only 70% of the labels, and then we look at the performance, the detection performance, and see how low can we go and still be able to get a good performance. So this is the weekly supervised approach. Yes.
Can you explain why it's so harder, the particle picking for tomography compared to SPR? Is it just the amount of data, or is there something else?
Yes, that's a good question. There's several things. So first, you have a third dimension, so everything is going to be more expensive computationally. Although, for picking, you heavily bin these tomograms, so they're not going to be that big, but still bigger than 2D. Then this is like a single particle type of sample, right? So you have your particles are uniform, and it's a monodispersed sample. That makes it easier. In the real scenario, like the image I showed at the beginning, everything is going to be very crowded, so it's going to be very difficult to even, annotate those images, right? And sometimes, you may, there may be things in there that you cannot see by eye, right? So how do you label those? So there's, those are like, some of the challenges. But the main, I think the main two difficulties are the crowdedness, and then also the fact that you have not just one type of particle, but you have everything it's in there. All the proteins are in the same volume. Yes.
Question, sorry. How can you have a pixel size of 1.3 something, if I recall correctly, with such a big pictures, like 5 by 5 holes? That means that we are talking about a square in which we have like 10 micron area squared, right?
Right. So
how can you have like such a perfect imaging, and then of course are you gonna crop and then perform like tomogram reconstruction?
Yes, that's in the end that's the idea. But I think you're touching on one important point, which is to get to high resolution you need small pixel sizes, right? But then you also want to cover enough area. So that's where the high throughput comes in, right? Now, without high throughput, you just won't be able to cover enough area to give you enough data to do high resolution sub-tomogram average.
Am I not aware of some cryo serial EM features? How can you do practically this with, for example, cryos?
You mean the data collection?
Yes. Like, is there like a script that you run and it's something that?
Oh yeah. This is these days the beam image shift is, I think it's even part of EPU or TOMO 5. That's from Thermo Fisher, and there's multiple implementations. But it's really easy. There's scripts for serial EM. I think that's what Pace TOMO does.
Because I was performing this for single particle, in which I do, for example, 4 by 4. But yeah, like I think at that point we had some trouble, so I had to do like 3 by 3, and so to me this was like huge achievement. Like
yeah, I know people do like 10 by 10, so you can really speed up things by using this technique, and it actually works in practice, and there's several examples of that. Yep. Okay, so then we say 70% is still a lot of labels, so we don't want to spend that much doing that. So then we took it down to 50%, 30%, 10%, and 5%, and look at the performance. And this is always, the more the better. But if you add these components, in the framework, you can actually get away with 10%. We think it's feasible, or maybe 15. But that's actually, if you only need to provide 10% of the labels, you're going to be very happy because you're going to have to be, you're going to have to do less clicking on these 3D tomograms. So we also ran this on this in-situ sample from Empire 10499. So these are the ribosomes inside the cell, and we actually kind of also label this manually and then reduce the number of annotations and see how with how few levels we can get away with and still be able to get good performance. And again, for 10% we pick most of the particles, and what that means, so the 10% is essentially we select about 10 particles on a single tomogram, right? That's what the 10% means in this case. So that's something that you can really do. It's not very time consuming, and you can really do it and then run it on the entire data set. So we have some metrics on the detection accuracy. When we reduce the amount of labels from 70% to 5%, and we see how how these metrics are actually changing. And obviously, when you reduce the labels, things get a bit worse, but it's still acceptable in terms of performance. And then we did ablation studies where we removed the consistency regularization and the positive unlabeled learning, and then we can see how that those components are actually very critical to the performance of this. So the bottom line is that we can train these networks using very small training sets, and meaning that we can train them faster and we spend less time doing annotations, and we can do this without giving up too much on accuracy. Yes, we're going to be taking some hit in accuracy, but it's not going to be that much. Okay, so now that we have, I only told you about one way in which you can get particles. There's many other ways. And I want to tell you a little bit about what happens after that, right? So that's the sub-tomogram averaging. At least that was the original term. Today, I think most people are actually doing this, not looking at sub-tomograms, but actually looking at the 2D projections from the TIL series. So essentially, what you do is this is showing a tomogram of these HIV viruses. And then you can select, in this case, a spike protein in 3D. And then you can actually look at the raw TIL series, and you can pinpoint back the locations of those images, 2D projections now. And there's a lot of benefits of doing that. And one of them is that you skip all the intermediate steps, like the interpolation, associated with doing rotation, 3D reconstructions. You do one step from raw data to 3D reconstruction. That helps you keep most of the high resolution information. You can actually leverage all the very powerful tools for single particle analysis that have been developed in this field for so many years. You can actually take advantage of all of that, all of those features. Obviously, in this scenario, because you're just looking at the raw data, there's no missing wedge. Essentially, you have no voxels where you don't have measurements because you're just using the data that you measure. And that means that you can actually do data-driven alignment, which is going to help you get to higher resolution. There is a caveat, right? So the caveat here is that each of those images are going to be very low resolution, much lower than single particle, because now we're fractionating the dose into 40 projections, right? So now each of those 2D projections is going to be much lower signal-to-noise ratio than what you're used to in single particle cryo. And that's actually a very important but, and I'm going to tell you now ways in which you can actually work around that problem. So many years ago, we came up with this constrained single particle tomography approach, which is essentially what I just told you. Instead of using sub-volumes, you actually go back to the 2 data. So here is a toy sample where I have 3 particles, P1, P2 to P3. And then I have a small TIL series with 3 tilts. So this is my first projection, second projection, and third one. So these are micrographs from 1 to 3. And then each of the micrographs, obviously, you will have projections from all the particles 1 to 3, right? And you could do this like in the regular single particle refinement framework, right? So in that case, you just cut out your particles here, and then you try to align them to a reference. This is using reference-based alignment. And the way you solve for those poses is you find you have the sum over all your particles. So I have n, not particles, but n projections, n particle projections. So in this case, it's going to be 9 projections. And then you're trying to find the poses that actually minimize this quantity. So this quantity represents the match between the individual images and then the projections of the 3D reference in that particular orientation. So you do that for all the particles, and now you find the angles that actually minimize that function, and that's what I call the unconstrained refinement, which is like your typical single particle analysis. What we did instead is actually try to use the constraints provided by the tilt geometry, because these are tilt series, and then instead of refining individual images, we actually take them as subsets, right? And we do that in 2 different ways. So here I'm showing the particle projections from I1 to I9, where I order them in this matrix, right? So a given column will represent all the projections on one particle. So this is P1, P2, and P3. And then the rows are going to correspond to individual microns, right? So this is M1. I have the projections of particles 1, 2, 3. And then what I can do in terms of refinement, I can actually do tilt series alignment, but now just using the signal coming from individual particles, right? So I don't need Gauss fiducials or anything else. I just use the signal coming from the individual particles, and I match to a high resolution reference, and that's going to allow me to actually improve the tilt series alignment using this approach. So the way you do that is you split the sum. So now you sum over the different micrographs or different tilts in your tilt series here, and then inside each term you actually sum only over the particles that are present on that image, and then the distance, the similarity function is actually identical. The images, that's the same. But now you have this G function here that is actually the one that imposes the constraints, right? So essentially, if you know that all those particles belong to the same image, when you're aligning your tilt series, you know that they all need to be moving as a rigid body, right? Because these are parameters of the image itself, not of the individual projections. So using this, you can actually improve your tilt series alignment, and you're actually using the signal coming from the particles themselves. The second thing that you can do is you can actually now, if you consider the columns of this matrix, you actually now have, so the green cylinder here is representing the reference, and now you have like a small tilt series that represents the projections of a given particle. And now you know that the spacing between them is going to be given by the tilt geometry. So you can actually refine the particle rotations by imposing those constraints between the different projections of that particle. And the way you do that is very similar. So now your sum runs across all the particles. So here 1, 2, 3, and then inside the sum you're going to have you're only going to sum over the particles that the tilts that correspond to that particle. Again, the same function as before with the constraints imposed by this G function. So this allows you to actually move the set of tilted projections as a rigid body, and then that way you can leverage the fact that you have the constraints provided by the tilt geometry, and that it would have some benefits, as I'm going to show in the next slide. So this is essentially the framework that I think most packages these days are actually using for doing high resolution tomography. So this slide has, I think, some very interesting things in them. And I'm going to go through, it will take some time to go through this. So I have 3 colors here. So I have green, blue, and red, right? So green is subvolume averaging. So everything that you see green, like this map is obtained from subvolume averaging, and then all these curves, they will correspond to subvolume averaging. So that's when you use the subvolumes, you align them in 3D, you average them, and so forth. So what I'm showing here is, this is the asymmetric triangle for this particular particle, which is GRO-EL, which has D7 symmetry. And these green dots here are actually showing the projection orientation corresponding to a single particle, right? So because this is a tilt series, they're all aligned, right? Because this is I'm rotated along an axis. So each of these dots is one of the projections orientations for one given particle. And then this D that I was talking about, actually, I didn't mention, but that's actually an iterative process where I actually try to minimize that in an iterative manner. And the x-axis here is showing the refinement iteration. So here we see that quantity actually gets lower as I'm being able to align the particles more accurately. And then that gives me some Fourier share correlation, which in this case indicates the quality of the reconstruction by sub-tomogram averaging. Now I'm going to switch back to the 2D images now, and I'm going to do the completely unconstrained refinement, right? Like the first component that I had at the beginning. I'm going to ignore completely the fact that these are tomographic projections, and I'm going to do the refinement. So if I do that, I actually get this map. Right? I'm actually able to lower that objective function by quite a bit, but the FSC actually stays the same. Right? So what happened here is I actually overfitted the data. If I look at the orientations of the blue dots after refinement, they're all over the place. So essentially, I just lost them completely. Right? So this is when where the constraints come in place and why they are so important, right? So now, if I actually impose the constraints, my objective is not going to be, it's going to be better than sub-tomogram averaging, but it's not going to be as good as the blue line, and that's because I'm forcing these guys to be on the straight line, right? So that's what the constraints are actually doing. So because of that, this actually helps with overfitting and actually increases the resolution. Right, so this is essentially where these constraints come in place, and they're actually really, really powerful. If you turn them off, things don't work because of such a low, signal to noise ratio of these individual projections. Okay, so as I mentioned before, because this is all done within a single particle framework, you can actually do very sophisticated things. So in single particle analysis, we do we actually have frames for each of our movies. And then we can actually track the movement of these particles at the level of individual images, and we can actually measure what is the contribution of individual frames to the 3D reconstruction. So by this, we can actually minimize the beam-induced motion, and we can actually compensate for radiation damage because we're actually measuring these quantities. And this plot here actually shows how each of the frames in the movie is actually contributing to the high resolution information in the 3D reconstruction. So here you see that the frames that are here at the top of this curve are actually the ones where you get most of the high resolution information. So because we're using this framework, we can actually apply this for tomography as well. But instead of having movie frames, we actually have tilts, right? So we can measure the contribution of individual tilts. And that looks something like this curve, where the x-axis indicates the tilt angle, the tilt angle from -60 to +60. And then you see, obviously, not surprisingly, that the best quality data is at the 0 tilt, right? And then, as you start tilting, you get the resolution lowering effect of the thickening of the sample, and also the radiation damage, because you're irradiating the sample, at every tilt, you're adding to the radiation damage. So we can actually measure this directly, and obviously we can use that for weighting this data for high resolution. So this is one example for one structure where you can see that most of the high resolution structure information only comes from the first two tilts, right? So the 0 tilt and then +3 degrees. Everything else is still very useful, but not for the purpose of high resolution. Okay, so these are some examples. We ran this on 12 till series obtained with this continuous tomography fast incremental till series, and then this was the original map that was published at 7 angstroms, and using this constraint single particle tomography, we were able to improve that to 4.8, where we can see obviously a lot more high resolution features just coming from the same data. And I want to point out that this data was acquired using continuous tomography, right? So continuous tomography, it can still provide better than 5 angstrom resolution, at least for this particular sample. So meaning that in many applications, this may just be good enough to speed up data collection if you don't care about going all the way to near atomic resolution. So this is just putting together, when we put together all the components like the tilted CTF estimation, exposure weighting, data-driven exposure weighting, and this is work that has been driven by Shanfu in my lab. We can see the progression of the resolution as you turn on all these different components, and this is for the DGTPA structure that I mentioned at the beginning for the BISECT experiment. Now, in the last few minutes, I want to tell you about this is a problem which is inspired, it's a biological problem. But the reason I want to tell you about this problem is not because of the biology, but it's because of the how it highlights some of the challenges that I'm going to describe in a second. So this is it's a retrovirus type K, where it's just like most other retroviruses. You have an infectious particle that actually attached to the host cell. And then the material gets integrated in the genome, and then it replicates itself, and then it buds out of the cell, and then you have this immature state of the particle that eventually matures and then goes back to infecting other cells. So again, I'm not going to focus on the biology, but essentially this immature state is critical. It's a key assembly intermediate. So it's important to know the structure of, and in particular, the GAG protein, which there are no structures available so far. So what we did is, in collaboration with Anna Sophia and Pei Jun from Oxford, Diamond Light Source, they produced these virus-like particles that have immature GAG, and these are 123 till series using the standard ± 60 every 3 degrees. And then here you can see one example of a till series of actually a tomogram, and here you can actually see the GAG protein, which is forms these lattices inside these BLPs. And here you can see a side view of that tomogram. You can tell that we didn't use ISONET. Right? Okay, so. The first challenge obviously was to pick the particles. That was, I cannot stress enough how big of a challenge that was. And then, so Yi Shu, he's actually in the room here somewhere. He actually figured out how to get these particles out. And just to tell you briefly, we actually detect the position of these virions, and then we have unsupervised segmentation in 3D, and this is using classical max-cut mean-flow algorithms, where you actually can determine like a minimal surface or like a graph cut in 3D, and you can actually get the surface, and then on top of that surface, you can just drop markers that are equispaced, and then that gives you your particles so you can feed into subvolume average. So long story short, we were able to get a lot of particles, and then we did like a sub-tomogram averaging initial reconstruction, and that gave us this map, which is around 7 angstroms. You can see alpha helices and everything, and then using constraint single particle tomography, we were able to improve the resolution to 3.2. Here you can see this asymmetric unit here in cyan, which is zoomed here, and you can see the alpha helix, you can see all the side chains, and that's the FSC plot. Now, this was a bit, it was hard to get this, but the main reason I wanted to go over this with this audience was because I want to point out the challenges, right? So these were 128 tomograms, right? So this is not 1,000, it's 123, but still. We had 8,000 viruses that these spherical features that we selected, and then we ended up with 240,000 particles, right? So, in single particle terms, 240,000 is actually not that much, right? We're used to doing like, millions, right? That's where you want to be if you really want to study like, heterogeneity. But this is, 240,000 for, for cryo-ET standards is actually quite high, right? Now, if you do constrained single particle tomography, where you actually go to the 2D projections, you're looking at a single particle problem where you have 10 million projections, right? So this is something that, the current tools can actually handle, but, it's going to put a little bit of stress because, there's a lot of projections that you need to refine, and then you need, even doing the reconstruction, right? If you knew what the orientations were, doing reconstructions of 10 million projections on a routine basis is actually not trivial, right? And we usually do this, 5 to 10 times, right? So you can, I think you can imagine, some of the challenges here. Yeah, so I didn't mention that, 10 million projections, just to give you an idea, the particle stack alone is 5 terabytes, right? So, just writing that much data is already going to take a lot of time, and I/O, you're going to spend a lot of time doing I/O if you don't do things carefully. Moreover, these tilts are actually movies, right? So I not only have 41 tilts, but now each of those tilts is actually going to be a movie. Normally it's 5 to 10 frames. It depends on how you collect the data. But that's going to add like another level, level to these 10 million particles. If you wanted to really do per particle frame, per tilt, per particle frame refinement, that's going to create a lot of challenges. Yeah, I think the main reason I did that is just to give people an idea of the scale of the problem and what the main challenges are. But I do want to close with a sort of like on a positive note, and I know I'm out of time, but this is my last slide. So I think I put this table together here where we have a single particle. We take one projection image, right? There's no angular range, no angular spacing. If you've been long enough with this community, you may remember when we did tilt pairs, right? So tilt pairs, we actually took 2 images, maybe one at 0 degree and one at 30 degrees. We did that for validation reasons and preferential orientation. But what you're seeing is that I'm showing this progression here, right? So all the way in the bottom we have tomography. So tomography is 41 tilts. That's the typical scheme. Then we cover a plus minus 60 degree range, and then the angular spacing is 3 degrees. But essentially what you have here is a continuum, right? You can either take one projection or you can take 41, right? So I think there's one example here that we did for BISECT where we took 25 projections, and then we have an angular range of plus minus 36, and then a spacing of 3 degrees. So I think the important takeaway here is that with these high throughput data collection schemes and these ways of analyzing the data that actually take advantage of the single particle tools actually allow you to navigate this entire range, right? And I think that's really where the power of this technique really is, right? So you can essentially decide on how many images you want, and then the tools are actually ready to be able to get you enough data and analyze that, to get you a high resolution structure. In practice, what it may turn out to be is that for different projects may require different schemes, but I think having these tools available is actually one of the important contributions of this work. So I think I'm gonna stop there. Again, thanks to the members in my lab and the work that I presented in collaboration with the Borneo lab at NIHS and then the Sang lab at Oxford. Thank you very much.